So I'm gonna now invite, as I understand, the Steve and Srinivas team. I guess, Srinivas, are you going to kick it off for us from Arizona, and with the report on BioFlora's approaches to control of HLB?
Yes, Pam, thank you. Can you hear me well?
Yes, wow,
Yeah.
look at you.
Okay, now that we are in separate rooms, there is no, the reverb thing going on.
That's great, go ahead.
All right. I guess I'm going to talk a little bit, but I'm making our first introduction. Srinivas, do you have the screen there?
Yes, I'm actually sharing the screen. I'm opening up the presentation.
Okay.
Srinivas, we're not seeing your screen share yet.
I'm looking at why. Hold on. Now I can probably, there it is.
So if you like click on your share. Okay, here we go. Here we go.
There we go.
All right.
So just a word before I speak. I think, which Jan mentioned about quorum sensing. I wanted to just mention a word about that. And Jan certainly made a very good point on quorum sensing and HLB virulence. What came to my mind then was, one of the hypotheses, probably we need to test this out, is that probably the difference between the grapefruit and the lemons, why you see the difference between, the susceptibility of these grapefruit or less susceptibility of lemons is probably the plant's defense mechanism, which is breaking this quorum sensing before it actually becomes virulent. That is one of my hypotheses. And probably that is one of the reasons our program is inducing the same mechanism in grapefruit to get, the plant enhancing the plant defense mechanism. I just wanted to add because that hypothesis came to my mind as soon as Jan was mentioning about quorum sensing.
Well, I'm sorry, I have to add, Srinivas, that when Steve first started telling me about what you were doing, I'm like, I want to know more. I want to know more. And then when I saw the presentations by Dr. Jones and I learned more about quorum sensing, I started connecting all these dots about it.
Very good, Jan. That's a very good point, Jan. So welcome everybody to this webinar. We are certainly, excited to present our over 5-year trial results, on the approaches to combat citrus HLB. And we are very excited to present. And it has really been a very fruitful and an informative day. Recently, Jan gave us on cover crops, which is actually a lost art. People should start, thinking more of these cover crops. And earlier we had about, biopesticides, biofungicides. Then we had a good deal about biocontrol on citrus. So it was a very informative day. And here we are, and we are very excited to present our approaches to combat, the HLB. And here I would introduce you Steve Pavich.
Hi, Steve.
Okay. Okay. Can you hear me now?
Yeah.
Okay. So I just want to make a little introduction here of myself and maybe my background on, citrus, and also primarily from table grape growing, but we grew citrus. We had 120 acres of citrus in the Terabello area of north of Porterville in the San Joaquin Valley. And they were, we actually grew cover crops, and that was that was quite a different routine back then, because at that time most people were using herbicides wall to wall, and cover crops and citrus trees was not done at all. And the biggest problem I had was just the skirting of the tree. Was once I got the skirting level just right, where I could get a mower in there, then it worked really well. And the shade of the tree took care of the weeds under the tree. So it really wasn't that difficult. And doing cover crops was not that difficult either, because you did mowing. I didn't do disking, it just did mowing. So my background again goes back into organic agriculture for about um, 45 years. I started farming organically back in 1971 after I graduated from Fresno State, and the first year that I farmed was a straight chemical triple 16, and just pour the commercial fertilizers to everything and just go for it. And everything just went wrong. I mean, the insect pressures were just incredible. Leafhoppers, Pacific spider mites, um, mealybug, um, and then we had various things like Eutypa dieback and other thing, nematodes. So it was really like all of a sudden you had to deal with all of these different types of problems, and you learn how to do it more with what I would call nature's method. So after Fresno State, it became a real journey in trying to study my soil and understand the chemistry of it initially, because the chemistry in the beginning is what we were all taught to learn about, as opposed to the biology or what the microbes were doing, even though the microbes were producing chemistry in their exudates. But so it became, we got to get the chemistry right. And then it was plant nutrition and NPK plus your trace elements, and make sure that your plant was healthy. So that's kind of my little bit of background. And we're going to go into some slides relating to HLB and the work that we did at Texas A&M in conjunction with the University of Arizona. And I think Srinivas, maybe just give a little background about that there. Maybe go on to the next slide.
Thanks, Steve. So Srinivas Makam here. I am originally a molecular physiologist and a microbiologist from Purdue University, horticulture. At Purdue, I worked on plant hormones and its molecular basis for signaling in plants. And in my master's, I worked on another plant hormone known as abscisic acid. And later on, after my Purdue, I came straight to Arizona State University. I was a faculty there, and I wanted to break off because, I wanted to do more hands-on, boots on the ground, connect the lab to the land. And that is what I started the Lab to Land program at the Integrated Life Science, Research Center. Currently, I am heading or directing the molecular department at the Integrated Life Science Research Center, which is a science division of BioFlora.
Okay, so about BioFlora and how my first experience was working with BioFlora as I was farming organically. Back in the early 70s, there weren't hardly any companies at all that were able to provide any kind of fertilizers to organic farmers. Back then it was composting and cover crops, and that was it. Well, it wasn't even composting then, because they was just putting manures on and cover crops. And the first compost that I made was in 1971 or 72, when I worked with a company called helped start, or called New Era Farm Service, where we actually made our own compost. And it was done based upon the Lukey method of compost making, and it was quite the science project itself. But then we needed other things, like, seaweed. I need some humic acid. I needed trace elements. I needed them to be organic. I didn't need them to be chemical. I needed to be able to feed the microbes. So that's when I came across a company called BioFlora with the founder of the company, Roger Blosky, And Roger was a Midwestern guy from South Dakota that came out west because he was tired of being cold and wanted to get hot. And of course, in Arizona, you can get hot during the summers. And he started a fertilizer company called BioFlora. And at the time, I worked with him and another gentleman out of the Tulare area called Ralph Juergens with New Era Farm Service. And I actually was one of the members that started New Era as a composting operation. And Ralph Juergens was the other gentleman that I worked with. And both Roger Roger and Ralph are on the other side now. They're kind of looking down here, but the continuation of BioFlora and where it's at right now has been basically in the areas of humic acid in the form of humus and microbes that are a collective part of the humic acid. So, BioFlora is a company that has a lot of it is a manufacturer of many different products, primarily humic acid, our algae products, our fulvic acid, and our seaweeds are our main products that we work with. And we work with sustainable growers as well as organic growers. And we've been doing that for over 45 years. As you can see here, this is our facilities, and I'm just showing you a little bit of facilities because it's a background on talking about HLB and what we're important, being able to get different kinds of products that are contributory to stopping HLB from being, taking over a citrus orchard. So, humic acid facility, manufacturing plant, microbial isolate production, fulvic acid, microalgae from our algae farm, seaweed cream manufacturing, blending facility, compost manufacturing, pellet mill, and Green Acres Research Farm. So we have a research farm that's actually in field, which I always stress that's important. A lot of times people do a lot of work in a greenhouse and consider it as research, but me as a farmer, I always used applied technology because that was the technology that actually was real, because it worked. If it worked in the field, then it worked. Working in the greenhouse didn't necessarily make it work in the field. So anyway, next slide.
At Integrated Life Science Research Center, which is the science division of BioFlora, this is a very unique setup for a company like BioFlora. We are probably one of the few companies who have all these departments in service to the farmer. Recently, we started the molecular division at Integrated Life Science Research Center. Here, the reason we started is we go down to the DNA level, and it has a faster turnaround and highly precise. And at the ILSRC, the science division, we have our own isolate production labs, and that is the beneficial microorganisms. Then we also have a microalgae laboratory, which is about 10 acres, here in the middle of the desert. And we are one of the first to introduce microalgae, in agriculture. So it has a very, very huge potential, and microalgae has done a great deal part of the program for HLB kind of work, which Steve will talk about later on. And we also have our microbiology laboratory, where we look at soil diversity, then do isolate production, identify the beneficial microorganisms. Then we have our greenhouse, where we have plant research in a controlled environment. So we have a state-of-the-art research labs, which is ILSRC, the science division of BioFlora. Just to mention a little more in depth of the science labs, what we have to the left in here, especially is the ELISA. So during my master's, I developed the ELISA technique to quantify the hormones, abscisic acid, And right now we are using the same ELISA technique to quantify, identify, diseases such as leaf roll virus, RNA virus. Well, it is detecting, viruses is another, challenge, especially when it is an RNA virus. It comes to another level of sophistication. So in here, we are able to detect and track such viruses and also PD, using ELISA technique and, phytochemicals. So we can, that is one of our in-house, competency. Then we have here the qPCR and the PCR technology, molecular techniques, where we can identify and also quantify, let's say, a pathogen at the strain level. To give you an example, Fusarium oxysporum, race 4, we can go up to the race 4, up to the race level, especially in lettuce crop, which is devastated Salinas Valley and the Yuma. Using these particular techniques, we can track and we can probe this particular pathogen, Fusarium oxysporum race 4, and other pathogens, such as the precision we have. We also can do other pathogens such as red blotch of grapes, botrytis. We have over more than about 250 probes we can look into. So those were the labs, and coming to our core technology, ours we have a very diversified core technology, which is in addition to nutrition, macro and micro. Our technology, diversified technology, consists of isolates, which is beneficial microorganisms, bacteria, and biostimulants. Broadly, we can classify our technologies into, of course, there is nutrition, then marine and microalgae, beneficial microorganisms, we call it isolates, then carbon complexes. Those are the humates. Go green, that is a diverse blend of 6 different microalgal species, which we grow in about 10 acres. This microalgae has a huge application, not only in soil health, but also the plant health in terms of rooting. Go green or microalgae is a huge source of the naturally occurring plant hormone, which is known as auxin, which is indole acetic acid. And this helps in rooting, and root development, and also the early establishment of the seedling. Then go green or the microalgae has a huge effect on the crumb structure of soil and also water holding capacity. It also adds carbon to the soil. I also want to say go green microalgae has photosynthetic capability, and this plays a huge role in soil carbon sequestration. That is something we'll have to look into how microalgae can enhance the carbon sequestration in soil. The seaweed cream is a marine algae from Ascophyllum nodosum. The main difference between our microalgae and what is existing as kelp outside is that ours is a whole plant extract. And secondly, we don't use potassium hydroxide to extract this seaweed cream. We use our the whole plant organic patented extraction process, and we fortify the seaweed cream with our cellular infusion technology, CIT. I will talk to you more on that. That makes the plant hormones in seaweed, auxins, cytokinins, and gibberellins, and more so amino acids in seaweed cream, more bioavailable and bioactive. Coming to isolates, Go isolates. Go isolates is a microbially activated, humic acid. This is an extract of leonardite. The base is Hu-mega, which is a carbon complex. And to this Hu-mega, we add, in-house, grown bacillus species at a very high, concentration. So in Go isolates, you not only have diversity, but also you have high concentration. Talking about diversity, recently we checked how much of microorganisms, how much diversity of these microorganisms are present in Go isolates. We found about 1,500 species. Yes, it's about 1,500 species present in Go isolates, which are naturally occurring. The reason why we have so much of diversity when we extract it is because we don't use harsh classic alkaline extraction, which is potassium hydroxide extraction methods. We have our own patented organic extraction process, which preserves this microbial diversity. And now you can see when you add this go isolates into soil, you are adding not only those concentration but also huge microbial naturally present microbial diversity. Isogreen. Isogreen is another great product. It is a blend of microalgae and humic acid, that is humega. And isogreen, I'll tell you, Steve especially uses this in table grapes, a lot of other crops as a finisher. What this does, isogreen, is it enhances the grapes crispiness and also the bricks, which is a total soluble solids, and also enhances the density of the cells. What that does is it gives it outcompetes things like issues like botrytis, and because of its cell density of the grapes, it has a very high shelf life. So these are some of the benefits of isogreen. C isolates. C isolates is a natural blend. What we do is we combine the high concentration beneficial bacillus species with seaweed cream. So now you have a very nice foliar spray, a combination of seaweed and also higher concentration of microorganisms, which is a good source of good biology, and also the goodies of the seaweed cream, which is plant regulators for a plant defense. IsoGuard. IsoGuard is water-based. And a high concentration of beneficial microorganisms, which is mostly also for foliar spray and also the ground spray. Coming to carbon complexes, to add on to these carbon complexes, we extract. We have a very patented extraction process where we don't use, like I said, potassium hydroxide, and hence we get our champion product, which is humega, which is a biologically enhanced or biologically activated humic acid. When we looked at humega, the amount of diversity present, there were about 1900 different species, and we are still counting, because there are several species still not identified. So when you add humega, you are not only adding humic acid, but also you are adding the soil biology. So another carbon complex is Fulvex. Fulvex is again our extracted using patented extraction technology, and in here Fulvex, the active ingredient is fulvic acid. Because of our patented extraction process, the fulvic acid molecule we recently characterized. We did something known as GPC, gel permeation chromatography, and we found that this fulvic acid is 10 times smaller than what is available in the market right now. That means it is 10 times more efficient. And also Fulvex, because of its extraction process, preserves the diverse nutrients from Leonardite. It has got about 65 different minerals, which is Fulvex, in addition to fulvic acid, makes this product also a potent electrolyte. And it has one very good amino acid, which is known as glutamic acid. Fulvex drives our cellular infusion technology. Diversity is a hallmark of all our products, and because of the cellular infusion technology driven by Fulvex and the nano molecule, because it is a very tiny molecule, very small molecule, and we call it as a nano molecule, and it is a very highly efficient molecule which drives the cellular infusion technology, and it works at the cellular level. We also use this fulvic acid for human health and also for animal health, and that's the reason our extraction process is food grade. And this food grade gives a very high quality even for agriculture, to be used along with starter fertilizer or with any other formulation, which to makes it more bioavailable. It is a natural chelator, so you can add along with your micronutrients or any other formulation, which will make it much more bioavailable, chelate, and takes it right through the cellular membranes. And in the human side, we did a small, bioassay where we looked at, its activity at the cellular level, and we found that it enhances the mitochondrial activity at the cellular, level. So that's the reason we call it as cellular infusion, technology. And at BioFlora, we started something known as Lab to Land program. So in here, what we do is we collaborate with USDA. We also collaborate with university for especially HLB. We collaborated with University of Arizona and Texas A&M University, and both AgriLife and Kingsville. They asked me to mention Kingsville, Texas A&M University, Kingsville. And in this lab to land program, we come a full circle. We talk to farmers, we analyze the samples, we see what is the problem, then we customize the product according to what exactly it is, what is the actual problem, and develop a program. So be it, we chilies in New Mexico, or let's say California table grapes, or corn, and soybean in the Midwest, or let us say, table grapes in Mexico or, South America. So we come up, full circle, for using this, particular lab to land program at ILSRC. At ILSRC, we've been working on citrus issues, for decades now. And the biggest challenge locally in Arizona, and also let's say in other places in citrus, is something known as Phytophthora. Then we also have something, another disease known as Antrodia species, which is, wood rot, which causes, wood rot. We kind of using the, our technology. If you see here, we used, polymerase chain reaction to specifically detect the Antrodia species. And, we challenge our, isolates, and we customized and, got, things under kind of out competing and enhancing the plant defense, over probably a year. We thought we had, kind of Antrodia under control. Here came, another problem, which was Fomitopsis. So that is the latest issue in citrus, especially in Arizona, which is known as, Fomitopsis. We are inching very close to a solution for, Fomitopsis. And I should also mention that when citrus trees, as HLB, all these pathogens, can be a, secondary kind of infestation. So when you use our program, I will talk to you about, the program. Steve also will mention about that for, HLB issues, enhancing the plant defense. You will also be addressing, these, other pathogen, secondary, pathogen issues. So our program, it has a, whole holistic, approach, enhancing the plant defense and also the, soil health. So it is not only, soil health, but also, the plant health, which Steve will, talk about this more. Can't hear Steve. I don't think Steve has his microphone on.
Oh, I'm sorry. Now I'm on. Anyway, these two pictures go back a long ways. When I first started farming, I could always look at the fields that had waxy appearance to the leaves and leaves that had kind of a dull cast to them. And what was the difference between them? The fields that always had the waxy appearance of the leaves were the fields that were the best quality, the best yields, and the fields that had the least amount of disease or insect problems. So it was always interesting to find out, well, what do you do farming wise that could go ahead and produce a waxiness to your leaves, and what's the big deal about it? And so I actually sent some leaves in because I wanted to know what the epidermal layer was doing. I wanted to know what the waxes were. I wanted to know what the thickness of the epidermal layer, which actually was more important than even the wax on the leaves. And when we went to our best fields and we sent them in an electron microscope, we found out that the parenchymal layer and the leaves, like if you look at the ones on the left there that had nutritional problems, those leaves had only a single parenchymal layer. The leaves on the right, if you took them under the electron microscope, had a triple layer of parenchyma cells. So when you go back to the waxes that are produced on there and what's the importance of that, insects, for example, thrips. When I went to the field and there was maybe weak spots where the leaves may have been adult cast, like the one on the left, and then where they were waxy or had the very strong parts, I had no thrip scars on any of the fruit that we were producing. And I was wondering, well, how was that? In fact, Jan, your father, Ev, we were looking at that. That was a little project that we were stumped on why that was so. And he actually found a thrip trying to lay eggs on a waxy leaf, and the thrip could not lay the eggs on the waxy leaf because it was too slick. And so the eggs couldn't attach to the leaf. Simple thing. And it goes back to, again, to the health of the leaves and the wax of the leaves. So these were the kind of things you discover as you go along the way, because you just thought it was like population dynamics. Because Ev and I would go back and forth about that. And I says, well, what about nutrition? You know, the guys from the Midwest, they always talk about nutrition, the organic guys that have been around it a long time, and says, all you need to do is have nutrition. You don't have to worry about bugs. And so we would go back and to try to understand, well, why do you have bugs and why don't you have bugs? So this was kind of an interesting one here in the waxes and how important that is. People don't look at. The other thing too, and one other thing there is, you notice how the leaves are flat. You know, you go out in a citrus orchard and you find that leaves are usually cupped at some point in time. And cuppedness means that their ability for photosynthetic energy from the sunlight in a cupped leaf is about half that of a flat leaf. A flat leaf, you get 100% photosynthetic ability of the sunlight to the leaf, which is very important. So it's important to have actually flat leaves. And, you know, people don't look at that one like, well, big deal. Well, it is a big deal. So that's another thing that we've learned through good nutrition. You get flatter, more relaxed leaves. Go to the next slide.
So coming up to our main topic, citrus greening, or one of our main focus, in the Lab to Land program is this challenge is, the HLB, which we took up about, 5 years ago. Working with, USDA and also the universities, our approaches is basically, a holistic approach. It is not a targeted approach and a program, based approach. It is very essential, to be a program-based approach, integrated approach, rather than, targeted, approach. And the nutrition is certainly a part of, big part of this program, which can go both in sustainable and, organic backgrounds. This program, involves both soil and foliar treatments. The program basically aims at soil conditioning, enhancing the soil microbiology, which is, starts right from the soil using our patented, microbial isolate technology and humates. This builds what is known as, suppressive soils. Building suppressive soil is a very long-term, effect. And what this does is it builds plant defense via enhancing the plant, immunity. To give you an example, one of our experiments in table grapes in California, when we tested, the plants under our program, we saw enhancement twice at least the amount of salicylic acid. That means the plant immunity under our program, is enhanced. The plant has a more, fighting capacity. This is very analogous, to building a healthy gut microbiome. So a healthy microbiome using your probiotics enhances your immunity. The same way our program enhances the soil biology, the rhizosphere richness and the diversity to enhance the plant defense. And of course, there is, nutrition, bioavailable nutrition, for foliar sprays. Our work with HLB, like I said, started about 5 years ago in collaboration with, USDA, Texas A&M, and the University of Arizona Banner Health Medical Center, which has the state-of-art and, contemporary electron microscopy, facility. This was under Dr. Day and Dr. Cromie. We used electron microscopy and something known as SIM, SIM. It is an enhancement of confocal microscopy. So we used both at the Banner Health, Medical Center, which does both, mammalians, humans, and plants with its contemporary, technology. And here, for the HLB effort in terms of collaboration, we did 2 phases. So initially, the phase 1 was a greenhouse, effort for about 6 months, and this was in collaboration with Dr. Yan Ping in the USDA Fort Pierce, Florida. So here we used, the grapefruit Duncan variety with Dr. Duan, and I'll talk to you about these results. And later on, we moved to our field trials, under Dr. Alabi and Dr. Mohammed Setamu, Texas A&M, AgriLife, and Westlaco and Kingsville, and then we had Dr. Wugo, who was at ILSRC, continues to also to work with us, and at the Banner Health Medical Center, University of Arizona, we teamed up with Dr. Day and Dr. Cromie, who have the state of art and about 4 decades of experience in electron microscopy. We did this work in grapefruit for over it's been about 3 years of field work now.
Dr. Makam, can I just add one thing to that, because I want to mention one of the things that happened when HLB was running rampant across Florida. This was around 5-6 years ago. I wanted to go back there and take a look and see what was going on, because there was one individual that was using nutrition in his orchard, and his trees weren't dying. Maury Boyd, he became very controversial because Maury was basically executive of a company and was doing really well, and the citrus orchard was just kind of a hobby for him. And but I had the opportunity to meet him and actually look at his orchard. I saw it firsthand and spent quite a bit of time. And Maury just says, I don't have a budget. I'm just going to throw everything except the kitchen, in the kitchen sink at the HLB and see what happens. And his trees didn't die. Well, with that introduction, I came back to BioFlora and I says, Roger, the owner, says, we need to look at nutrition and study it a little bit closer and understand of maybe providing more quality products of the actual nutrition itself. And this is where we started what was the original thought process, what we did, and then we got an experimentation here and was successful at what we went forward with. So Dr. Makam, back to you.
So this was, our USDA Fort Pierce, Florida, Dr. Duan's, Yangping Duan's collaboration. It was a 6-month, experiment with, BioFlora program involving both nutrition and also, micro and macro, and also iso green, which was a combination of, Go Green microalgae and Humega, which is microbially activated, humic acid. So initially, we could, using qPCR, technique at the DNA level, we could detect, initially to start, but at the end, we used about 6 programs. I'm not giving all the details of all the 6 programs for the, time, issue. But the program number 6, at the end of 6 months, we could not detect any Libribacter. So Libribacter asiaticus, which is known as which is citrus, greening causing, bacteria. So nutrition The boron zinc, foliar, spray of boron zinc is very essential, as one of the micronutrients, because boron is certainly essential when there is, this is library bacter. I will show you how it actually clogs the phloem sieve tube. And this boron helps to overcome any kind of let us say source to sink movement of the sugar. So it also helps in sugar movement. And zinc, of course, for plant immunity and also for enzyme activity, which helps in plant defense. So nutrition and also biostimulants and microbials also in about 6 months in greenhouse gave us no detection of this library bacter completely. But is this enough is the question. So can we translate these greenhouse trials to field? No, there is another environmental component to it. That's the reason we head out to the field in the University of Texas A&M University Agri Life, and we gave about 4 programs. We gave color code for easy identification. Everybody likes colors, so we gave program 1, which is red. This is mostly, like Steve mentioned, mostly nutritional, but there was a little biostimulant and also a little bit of microbial. But program 2 also had macronutrients, microalgae, and go isolates. But in here, the micronutrients was a foliar spray. Program 3, which we gave as a yellow, color coding, the only difference was iso green, which was a combination of microalgae and humega. Mind you that this is the program which actually worked in the greenhouse. Program 4, one of the main differences is that we added seaweed cream and our cellular infusion technology, Fulvex. So this was the main difference. The USDA guys liked seaweed cream because it is a very good source of amino acids and plant hormones. And the white, of course, we gave for controls. You see, initially the control plot, the white, you can see how much devastated it was when we took up this particular experiment. And the library bacter, Asiaticus, LAS for smaller short form. What this does is it clogs the phloem sieve tube in the veins, in the midrib, and also in the petioles. And finally, what happens to the leaf? It is deprived of all the nutrients, and also the photosynthesis coming on. And what happens is this kind of yellowing, asymmetric yellowing. And finally, what happens is the leaf finally succumbs and defoliates, and this basically over time the entire tree collapses. But how does this last bacteria look inside the phloem of the grapefruit? We were very interested to see it, and we went straight into looking into the electron microscope. So these are the first glimpses of right into the phloem of the sieve tube of the grapefruit. This is a library bacter Asiaticus, which was looked at higher magnification, 43,000x and also 60,000 x. It is a typical rod-shaped bacteria and inhabiting and clogging the phloem. We tracked this bacteria throughout the program. And what happens to this particular program, and currently what we are also doing is we are also doing something known as immunotagging, immunodetection of this particular library bacter to further follow this bacteria throughout the program in the phloem. So where does actually it kind of populate? Where does it prefer? How does it colonize this particular leaf once the psyllid infects the leaf? So we looked at the various levels during our program 1, 2, 3, and 4, which we have color coded. We were looking if there is any clearing or if there is any differences in this particular program using qPCR. So it is a little not that complicated. I can tell you this much. qPCR gives the CT values. These are known as threshold values. Higher the CT values, lower the infection. So that means if the values, if the bar raised is higher, that means your library bacter is lower in in the leaf infestation. So if you look at this at 16 months, at about mid range of our trial, you can see here both the red and the orange treatments had very high CT values. That means the infestation compared to the white and other treatments had gone down. But when we looked at the rating, tree health rating, the orange one stood up. The orange one stood up, and it basically said that hey, this program 2 or the orange is doing something. Then we continually followed this particular bacteria over the years, and we wanted to look further and using the transmission electron microscopy for the leaves at the petioles, midrib, and also the veins at higher magnification. So this is what we found. So we found this is the midrib. If you look at this, that's the last bacteria. To my astonishment, we found at least more than 21 different bacteria, in this grapefruit. And this is the veins. And in the PDL we saw, but we did not see much. We scanned through. Looks like the bacteria is preferring the midrib and the veins over the petiole, at least in the initial infestation, actively infestation times. Chances are that later on over infestation, it might basically spread and move towards the petiole, and finally the leaf might abscess. So that is basically the disease and succumbing to the disease. But during the active infestation period in grapefruit, we kind of saw that the bacteria prefers the midrib and also the veins. But in the program, what happened in the program? So if we look at the program 3, 1, 3, and 4, the library bacter certainly had come down in all the 3. But in these 3 different treatments, we looked at the genomics. I will talk to you about that later. There seems to be a different mechanism in either blue, in either red, or blue or yellow. Although there was decreased, microbial load, in these phloem sieve tubes, the mechanism of this program seemed to be different. We were curious. Do we get anything where we don't see any, bacteria at all? It did. To our surprise, in the orange treatment, T2, we did not see any bacteria in the phloem. So the phloem was totally, flushed out. And we were, astonished, and we screened through a lot. We screened through a lot of, leaves and a lot of sections. We could not, see any particular, bacterial infestation. So the question here is for our future research: is this a new phloem? Is the program kind of triggering the grapefruit to regenerate new phloem? This is the difference between the grapefruit and the lemons. Like we were talking about phloem sensing, quorum sensing in citrus or the lemons. One of the reasons why it is not that susceptible is because it has the capability to regenerate the phloem cells very fast. But in grapefruit, it doesn't have that capability. Looks like our program is triggering that defense mechanism. So that is something we'll have to look into the further focused research. So we were excited to give or to see these particular results. Our program is a blend of all these goodies from all these particular treatments or particular program. We all put good elements of all this program, and Steve Pavich will talk about this program as one whole entity. So we looked at genetic level. So we wanted to look at a little more what is happening at at the gene level, at the gene expression level. So we looked at gene expression using something known as RNA-seq, and this RNA-seq gives you the genes expressed as a result of your program. So this is known as the cluster analysis. And the red means the genes are going up, and the blue means the gene expression is going down. Here in this panel alone, in the control, there are about 1200 genes of the entire grapefruit. In other treatments, you can see the variation of the genes as a response to these treatments. But look at these two. The best, one of the best programs where the entire phloem was flushed out. Wherever there was enhanced expression, exactly there is decreased expression. Wherever there was decreased expression, there was enhanced expression. That means the program turned the genes completely around. But when we looked at another unique analysis, we found only about 100 genes in this particular treatment, which was responsible, very unique. Probably those 100 unique genes, only 100 for such kind of an effect. So we are identifying this unique pathway, where the plant defense is enhanced to overcome this HLB particular issue. We are very excited to decipher this pathway, metabolic pathway, and we are very close on deciphering this along with USDA and University of Arizona. So here is at the field level, recovery and yield. You can see at the end of about 3 years, there was really a turnaround of the program. At the beginning, the trees were really devastating, but right now we could basically see the trees were turning around. But this is you don't see any fruits because the pictures were taken post harvest. And those post harvest means what happens to the yield. So what happens to the number of fruits or the total yield. When we looked at that, the total number of fruits per tree in both orange and the yellow ones were higher than the previous year. That means there was at least 80% enhancement in the yield in the orange program too, and also in the yellow ones. So these two really stood up in number of fruits per tree. But what about let us say fruit weight per tree? Again, this orange and yellow treatment showed up the most fruit weight per tree enhancement compared to the previous year. So there was the program really turned around. This is let us say number of fruits in terms of yield, that is quantity. But what about quality? So we looked at in grapefruit there are sizes, different sizes of the fruits. The biggest one is known as the jumbos. So if you look at the blue, this time the blue program gave the highest jumbos, but the orange one also gave more than blue. So there is an enhancement of not only quantity but also the quality. So here it is, the whole program. That is the nice thing about the integrated approach, a holistic approach. Not only it enhances the plant defense immunity to overcome the plant infestation, but also it gives the plant the power to regenerate, recover, and also give more yield in terms of quality and quantity. So the program what we speak about gives you the blend of all the goodies from all these particular program. So we had 4 different programs giving 4 different effects. So Steve Pavich will talk to you about these programs where we can put out, farmers can use it and really turn things around for this infestation.
So when you look at actual sustainable programs, one of the things that, and I'm going to talk a little bit about quorum scenting before I get into the understanding of what developing a full program actually means. And that goes back many, many years when I worked with in table grapes to understand what was going on microbially in the soil. And this was back in the 80s, because there was nobody who was able to do testing of the soil to find out what the microbes were doing in relationship to nutrient uptake. So I went all over the place looking for labs, and I found a lab actually in my backyard, Phoenix, Arizona, with a young woman called BBC, Vicky Bess, and she had a lab that was called BBC Labs. And I went to her to try to get her to develop a program that I could identify at least different speciated microbes that were going on in the soil. I told her what I wanted to do, and I wanted to understand the relationship between the microbes in the soil and disease in the soil. So at that time, I was fortunate enough to actually, out of the 13 different farms we had between Arizona and the San Joaquin Valley, one of the farms was actually a suppressive soil. And at that time, there was a young professor, researcher that was doing work at Cornell, and his name was Dr. Klepper, and I think it was Cornell. But anyway, he was trying to understand what a suppressive soil is and how its relationship to growing crops. That, you know, there just was about 3% of the soils where diseases just didn't happen. And insect diseases and problems and bad insects also didn't happen, at least in numbers that weren't damaging to the crop itself. So that got me on an endeavor to try to find out what was going on in the suppressive soil that was taking place on my one farm north of Bakersfield, California. We called it the Freeway Ranch. And the Freeway Ranch just never had any high leafhopper populations, never had Pacific spider mites. There weren't any mealybugs in there. They didn't have, they had a few nematodes, but they never were out of place. And just, there was nothing that ever happened to that field. It was just on automatic pilot. So that was trying to understand, well, what was it that was taking place? At the time, and you learn things by accident sometime, and it's a little story about how did I find out that something different was taking place in that particular field as far as the chemical structure of what was going on in the physiology of the plant. So we were doing testing at the time to claim no detected pesticides in our plants, and we had this NutriClean program, and so there was not any detected, pesticides in our plant. Yes, we were organic, but we had non-detected pesticides. Well, it turned out that we would test the leaves and we would test the fruit just to see the difference between the two. So we were able to make a substantiated claim that it was not in the fruit or wasn't in the plant at all. But up came one of the ranches, and when we were doing the testing with National Food Laboratory up in Dublin, California, they detected a chemical that was deemed a pesticide, but it was an unknown chemical. So we said, well, we're not using any pesticides. Where did the pesticide come from? So the pesticide was an unknown chemical compound, and they named it chemical 413 on the spectrum of the way that chemicals are actually named, and they called it 413. And I said, where did that come from? And he says, well, it's an unknown chemical, and but it's only in the leaves, it's not in the fruit. So the transport mechanism was different, and how did it get up there? Well, we went to the soil and we tried to identify what were the particular microbes that were in plentiful, numbers in the soil. And what we found out was that there was a particular species, Pseudomonas putida, that was in there, and it was actually producing that chemical in the breakdown of different nutrients in the soil in its exudate. And that the mode of how it traveled up into the plant, it only went to the leaves, did not go to the fruit. So that was just an example of one particular microbe and we documented this all, and I was just amazed at how something like that could happen, and how this particular farm, which we called a suppressive soil, this was one of the components of making that soil a suppressive soil. Now, never mind, we had cover crops at that time, and that field happened to be flood irrigated because it was not, we didn't actually own the farm. We leased the farm, but we leased it for, 40 years. And, the owner didn't want to put in drips, so we had to work with it, flood irrigation. So we were able to do cover crops every year and work with it that way. So going back to trying to, again, understand that, BBC Labs and Vicki Bess she was really, I said, why don't we just find out what's going on with the population of microbes? Because this was like an important thing to me. I says, what microbes are going on in December? Which microbes are going on in July? Are they different? And is the population highs or lows? And anyway, when we worked with her, we found out that there was all kinds of different numbers. And for example, there was a Pseudomonas syringes which would only occur during a 3-week period in, if I remember correctly, in April. So when we had things like that were occurring, it was just really, really difficult to unravel what was taking place when we wanted to understand a monthly of microbes. But what really happens was in the top 3/4 of an inch in your soil, most of the activity is taking place there. And what we found out is that the pH of the soil in that 1/4 inch, because the microbes would proliferate so heavily, they would give off CO2. And then when the water would hit the CO2, they would form carbonic acid. That CO2 was very important in the acid production of the soil by the microbes, so that you were able to you push salts away and all kinds of other things that were problematic. But when the soil dropped to a 5.2 pH, the population of microbes declined because there wasn't enough oxygen in the soil to sustain them at those high levels. And as soon as their numbers went down and then the pH came back up, they would start populating again and they would go ahead and run through this cycle. Well, we found out that when we put limestone on the soil, just at a couple of pounds per acre per week, we found that we were able to adjust that pH and keep it at a 7.0 and keep the population of microbes at a very high level. And at the same time, they were able to produce the carbonic acid that was necessary for us to push salts and other things, and also solubilize some of the nutrients that were more insoluble in the soil as you put them in. So by having that whole little phenomenon take place and trying to understanding that, we learn things as we go along. But it was important to note that the studying of those microbes on a monthly basis and understanding their population levels was the most critical thing in trying to understand how we can keep their populations high and keep them sustainable and keep the pH from adjusting up and down due to their production of carbonic acid. So that was the importance of limestone, not gypsum, but limestone. So this goes on to looking back to this macronutrients, micronutrients, mineral input, going back to Maury Boyd on the types of things that he used in his particular programs. And I think the slide actually explains itself how there is diversity of inputs is very important. And it's not just putting on NPK and trace elements, it's having a carbon source like a humic acid and pure humates, which is not having undigested organic matter where you have different types of microbes in those environments, but pure humic acid to sustain the mesophilic bacteria that you want to have in your soil, or microbes that you want to have in your soil. So why don't we go to the next slide? When we really get down to the end of looking at this, and I know that it's a bottom line issue that when we looked at Maury Boyd's trees, this was like 6 years ago, his trees were alive. They weren't particularly super healthy, but they were alive and they were getting production and they met the quality standards, the minimum quality standards that were necessary for him to sustain himself and stay in business, as opposed to his neighbor's dead trees. And that was what was just so evident, that that Maury didn't have an agenda. He wasn't a scientist or anything. He just said, just pour anything to it and see what works. And what worked was a full nutritional program that was able to allow the trees to stay alive underneath the extreme press of HLB. So, so practicing a BioFlora program goes to the next level, we feel, because there's so much more to the density of our products in the form of humus, in the form of the algae and algae products that we use, which are all beneficial, and ultimately in the efficient uptake of nutrients from the soil up into the plant. And that's like one of the most important things, because a lot of times you may have those things in your soil, but they have difficulty in breakdown. They have difficulty in getting up into the plant, and that's and that's where the HLB management of soil and foliar isolates, biostimulants, nutritional programs will improve and maintain plant health and enhance that plant immunity while improving the economic production. So you don't have to sacrifice yields. You don't have to sacrifice quality. In fact, it becomes enhanced. And that's what we were able to do in our approach. And Dr. Makam, maybe you could give your strong message here on our technology in the last part here.
So the bottom line, like Steve emphasized, it's certainly a holistic approach. It's a integrated approach towards enhancing the soil health, and also building a very strong rhizosphere, and also doing the nutritional program, not only for ground and so for foliar, but it's a holistic isolate and let us say biostimulants, which enhances the plant immunity, not only to overcome such infection, such devastating infection, HLB, but also to produce and also to give yield with both quality and quantity. It's a program. We do hope that you got these programs, and please also get in touch with us for this particular program. We'll be more than happy to assist you and to guide you through this particular programs, rates, and applications.
Wow. I'm eager to hear some questions from the panelists. I don't see very much happening. Our attendees number has shrunk by about 20%, but this is getting down, really drilling down into what's happening in this at the cellular level. And so I'm just, there are people hanging in there who I'm sure would like to ask you some questions.
I have a question. It looks like if this picture that's up right now is the orchard you're talking about, it looks like he's fairly conventional in all his other practices, not a growing cover crop or anything like that, and maybe herbicide sprays. So what about his other practices? It looks like you guys are really onto something with this. This was an incredibly sophisticated presentation and technology that you're doing.
No, this is not a ground. This is just a representation of the citrus. This is not a trial ground. Sorry about that.
Right, but so tell me about his practices of the guy who kind of developed the throw everything at it, and he's got improvements, and then it looks like you're doing some improvements also, but what are his cultural practices?
Well, Maury Boyd was clean culture. If you go from southern San Joaquin Valley on the east side, where most of the oranges are, you can find an occasional organic orchard where somebody may be using cover crops, and I think it's a loss. It's something that the farmers are not looking at. Managing those cover crops is a whole other ball game. Mowing and being prepared to be able to do those systems is something that larger citrus growers just are not set up to do. So we're trying to accommodate them without changing their whole system. Although I'm pro cover crop, I'm pro organic matter recycling, I'm pro that makes everything work better in a live soil system. Although carbon sources are hard to find in that respect, and that's why the humic acid is such an important component and making these things work. It gives a house for the microbes to actually live in a barren waste desert. So
Right.
that's the hard part of what we look at. And again, you can go ahead and beat your head against the wall and say, you need to put a cover crop in, you need to do this, you need to go ahead and do all these things. But at the same time, this is what they're doing now. And eventually,
Well, if you don't compromise, you won't work with very many people. You got to work in. Yeah.
No, I agree with you. And I'm just saying that we're trying to educate as many folks as we can to the plus side of looking at the plant nutrient uptake efficiency a little bit differently from what they've looked at from just NPK inputs.
Right, right.
This is Jim Davis. I have a question too. It seems that when a citrus tree gets infected by HLB, the branch that initially gets infected shows more viral particles than other branches. In other words, it takes a while to spread through the tree and may even have to be spread by the insect to different branches. And I'm wondering if that was accounted for in the counting of the particles in the leaves and the midrib and phloem cells. Did you sample leaves from, the same branches over the course of that trial, or were they taken from different branches, or how did you account for that variability in the presence of the tree of the viral particles? Thanks.
Correct. So it's a bacteria, it's not a virus. So yes, you are right. Initially, when the actively growing leaves are highly infested later on, and also there is something known as flagella. So these bacteria, there is a recent publication saying that when the psyllid is infecting the younger leaves, the bacteria still has the flagella. So, and over the course of time, some evolutionary mechanism, it doesn't need it. And it basically loses the, entire flagella. So we looked at, these particular, leaves, which we looked at. We wanted to see if there is any flagella or not. There was not flagella completely. But when we looked at the younger leaves and the older leaves, there was certainly a little difference on, the kind of colonization, the kind of preference, and how it basically does. What basically happens is over time, and this was also one of the effects of the program. What happened was in one of the programs, RED, the programmed enabled or pushed the bacteria out of the leaf. We saw that more in the PTO. So that is one of the defense, particular mechanism. Chances are that as the, as the infestation goes in, the whole leaf get infestation, and finally it, upsizes. So what we saw right now is the active initial infestation period, where it basically prefers the, veins and the midrib. One of the reasons. So I was thinking of why, and the reason is it is the main sites for the movement of photosynthase or the sugars when the photosynthesis occurs, and that's one of the reasons.
At the University of Florida, they've been doing numerous studies along these lines, looking at, especially since Maury Boyd brought it up, but long-term 3, 4, 5-year studies where they've looked at various micronutrients and looked at various positions on plants and done yield studies, and they've done chemotherapy and thermotherapy and done phosphorus acid. And the micronutrient aspect often is just one small part of the whole program. And that a lot of the problem is down in the root system. Jim Graham has been looking at the impact of HLB on the below ground, and it's, you know, that's where the roots are dying. And you know, it's reflected in the canopy. And, you know, a simple, I'm sorry, I didn't mean simple, but, you know, taking one approach is not going to address this. It's going to be more than micronutrients.
Exactly. You know, when we spoke about, when we actually went to Florida, we spoke to, you know, farmers and discussed our, kind of, approach. They really said we tried, modified, but that's not enough. You know, I completely agree with you. They said that nutritional program is not enough. It doesn't have just enough juice. That was our program one. I did not label it as modified cocktail for this reason. That was basically nutritional approach, and that's the reason we always say it is more than nutrition. Nutrition is certainly important, but it is more than a nutritional approach. It takes a whole program. It basically takes a whole program, and over time to build the suppressive soils, or say they enhance the plant defense mechanism. But once you achieve that, you shouldn't be stopping, because these bacteria is they are opportunistic too. So once they find any kind of a slit, they will particularly get back. So you need to be really persistent and consistent, not only with the nutrition but also with other elements what we basically looked into the program for enhancing the plant defense, but for roots, for ground, and also for foliar. It's a whole approach. I completely agree with you.
Yeah, and let me add to that. And I would like to say that, when we were back in Florida, you looked at the ground. It was like blow sand. It was like a sandy beach. And, the farmers that were there were, using NPK fertilizers, and it was working for them at the time until, of course, they would get any diseases or anything would come along. And then next thing you know, citrus cancer, for example, just devastated the state, almost as bad as HLB. And so when you look at their soil, that just, less than 1/4% organic matter, probably 1/8 of a percent, doesn't hold anything. Microbes were not living in that environment at all. And they weren't looking at the microbes at that time. And so I think this is an important part of the understanding of how these work. It's not just pouring some chemicals on the ground and saying, I got all the trace elements. No. It's actually having the microbes be part of the central figure of how those nutrients actually get up into the plant. Okay.
So how much is the program that you're proposing, the Orange program, cost per acre, and how much does it cost to replace an acre of citrus? What's the trade-off there?
Well, the cost of programs vary depending on the grower themselves. There's some growers that want the highest yield and highest quality, and they don't care how much money they have to spend to get to that point, although they do. They're always on budgetary concern. But the good growers are going to spend $500+ an acre, let's say in a program, $450 to $550 an acre on foliar nutrients, soil application of different things. The lower budgets could be anywhere from $350 to $450, and then there are a few growers that actually get up into the $550 to $650 range, because I deal with them all, and I know that for a fact. Now, on the other hand, what does it take to do a very robust program, a first-class program that is going to suppress the HLB and keep you out of the problematic zone of getting HLB? We figure that it's $100 to $150 an acre in addition to, let's say, just a regular medium tier type program. And at the end of the day, looking at trees, like when you go into Florida, entire orchards that were devastated, nothing's alive, or half the orchard's alive, or they're getting half yields, or the quality is just terrible. And, it's sad to drive around the country, in citrus country. So programs are dependent on keeping out of the problematic area. Could be an additional 100 an acre to maybe 150 an acre, so that you stay clear of HLB. And that's really nothing in comparison to loosening your trees.
And do you think that program would be preventative as well as remedial?
Yes. The thing about when you have a humic acid that has over 1,900 species of beneficial microbes to it, you begin to build your soil in a much different way of diversity. Because, you know, there are people who say, oh, I got these 7 microbes that are going to suppress a particular pathogen or disease. 7 microbes. Well, how about 1,900? How about 19 million? 97% of the microbes don't even have a name yet. 97%, or at least some microbiologists told me that one time. I don't know for a fact, but let's put it this way. We don't even have names to most of the microbes that are in our soil as we speak. We know what groups they may belong to, but when we actually go ahead and DNA, structure them out and find out, well, what species are you and what's your name? We don't even have a name for them. So we're constantly learning and trying to understand how important diversity of species in that environment is. And so the more diversity of microbes, that's beneficial, the less problematic your soil can have. And in the suppressive soils, we found a tremendous amount of diversity, not necessarily high numbers, but diversity was the most important single thing that was in those soils. The other thing was silica. You say, oh my gosh, well, what important nutrient was it that was really probably one of the most important nutrients? It was silica. And I say to myself, well, okay. And I looked at my 13 different farms and which ones had the best yields and quality and everything. The silica always was the highest in those fields. So there are some components that are necessary, but diversity of species, I think, is the most important one. And it's the one that gives you the insurance policy that somebody in that group is going to be able to take a pathogen down when it's necessary, when they're called to duty in the corn sensa arena.
When I thinking about the micros, there's high boron in our county soils, as I understand it, and I think I recall Srinivas mentioning boron as being protective.
Yes, so that's one of the micronutrients which is essential for sugar translocation. So what this does, HLB, is it, as you saw, it clogs the phloem sieve tube and blocks the sugar translocation. But boron, what it does is it tries to compensate, at least not completely. That's the reason nutrition is just not enough. It tries to get the sink and source relationship continuing, while the rest of the program helps with plant defense. So it's a combination.
Yeah, one of the things to remember here is that Florida is Florida, New York is New York, Washington State is something else altogether. And California and Southern California is different from Northern California. We have boron toxic levels here.
Yeah.
And, so whatever it is, nutrition is going to have to be adapted to the environment and the soils. And a commonality to most soils, and again, Florida may not be soil at all. It's just crushed coral, I think.
And yes.
But most soils are iron, aluminum, silicon, and oxygen. That's common chemical formula for them. And so you really have to find some weird situations where silicon is lacking in a soil. And then the other thing is that HLB, these micronutrients are probably just masking HLB. Jim Davis said, remediate. You're not going to remediate a tree that has HLB. It's an infection. It's like, it's a disease. Coronavirus is a disease. It's into the humans. It's into plants. And they've done awful things, with antibiotics and things, and tried to clear it up in trees. And
Yeah, but Steve,
Well, was I'm
masking what's in there.
the
if
actual
I'm mistaken then in the presentation, I thought that they said that the data in the orange one showed that they had cleared the bacterial particles from the plant.
We don't know. That is what, whether it is a new phloem or whether, it is the same phloem. That is the question. So
Okay.
it can be a new phloem which is, come up. That is our next approach to find out.
Okay, thanks.
So I agree with Ben Faber that, recently, I kind of got hold of that, paper where they literally, injected antibiotics, to the whole plant. How about your environment? To how many trees do you go, inject, antibiotics? How are you going to? The whole challenge in that case is not only environment, but also your mode of application. So how are you going to, apply to so many, trees and, antibiotic approach? So that's another, challenge taking that, particular route.
Mhm. Yeah, I
There
invite.
is a way to do that, by the way. They did that with paradecline and paracilla, which is a very similar situation. I won't go into it long, but there is a way they did it with milk jugs and terramycin and one tree at a time on 1,000 acres. And then a University of California guy invented a machine that injects it into it. So I'm hoping we don't actually have to go to that.
No, gosh.
Well, you have a better solution. That's what, our 5-year research is all about.
Right.
Steve Sprinkle, did you have a question or a comment?
Yeah, I did. I've really appreciated the comments, and thanks for answering the two of the questions that I had previously, one on the cost per acre and also on the element of prevention, because here in the Ojai Valley and principally in Ventura County also, we're looking at a prevention program rather than a cure program. And I think that it's good to know that there are answers that you can provide to people who are interested in alternatives to the regime that we're going through right now. I think Ben probably could know this specifically. I think that so far in the southern region, we've only found, I think, a little over 2,000 trees that are actually affected that have been removed and destroyed. But of course, that's going to be, that's indicative that there's more of that coming. And so I think that it's good to have a kind of a comprehensive and very believable scientific approach that that doesn't make promises that haven't been proven and haven't been that are sort of just full of hope rather than science. So I'm really happy to see this as an alternative and something that we can explain to people who are interested in transition.
Thank you.
Well, I'd like to make a comment about that, because I think it's very important that, I'll give an example: Pierce disease, which affected table grapes back in the early 90s, and we actually had a field that was had 4 vines that died from Pierce disease. And, I was told that the entire 400 acres would be dead within a very short amount of time, within a couple of years. Well, it never happened. And again, we went back to our nutritional side of understanding that. In the meantime, there was a tremendous amount of money spent by the university in studying Pierce disease and trying to get a solution. It's not a hit on the university. It's just that sometimes when they look at these programs, it becomes long-term programs that don't necessarily end up with a cure or a solution. At UC Riverside right now, the $8 million is going to go into HLB. Well, if somebody discovers a solution to that before the $8 million is initiated, should we forget about studying it, or should we continue studying it and talk about that there is no solution, or there are multiple aspects or ways of looking at the problem? And I think this is something to where, it's hard, especially if you're involved in the 8 million going to UC Riverside. So again, I've certainly Certainly, and I'm not trying to bash university. I'm just trying to say that when it comes to research, the adjustment of the research is very difficult to adjust, especially if something is discovered like this that points a different direction in the solution.
I would agree with you, Steve. It's hard to adjust, and like I said earlier, we need some immediate systems integration, and the university is set up for long-term research, rather single factor analysis, rather than what you're doing.
Amen.
Good.
Yeah. Amen there.